So I'll just get started. And today I'm going to share my recent research about synonymous mutations, and it's an ongoing manuscript. So I'm going to first take a few steps back to look at the core of molecular biology, which is the central dogma, the idea that the DNA is transcribed to RNA and then translated into protein. And this framework gives us a clear path for understanding traits and genetics disease. So when a mutation changes the DNA sequence, it leads to a different amino acid in a protein, and it can directly affect how the protein folds function or interact with other molecules, and that leads to disease. A textbook example is sickle cell disease, which is caused by a single non-synonymous mutation in the HBV gene, and that one synonymous mutation leads to one non-synonymous mutation leads to one amino acid substitution. It distorts the shape of the hemoglobin, causes it to aggregate, and leads to the disease. So this idea that the mutation can change protein sequence and disrupt the function is deeply establishing our understanding of genetics and diseases. But today I am going to show you that this isn't the whole story. And specifically, we're talking about synonymous mutations. Those are mutations that change the DNA sequence that do not change the encoded codons. So the protein sequence stay the same, and people would assume that, oh, the structure and function is the same, and synonymous mutations are usually viewed as functionally irrelevant or neutral. And this assumption that they are neutral actually shaped the foundation of how we understand biology at the molecular level. So this assumption allowed synonymous mutations to serve as a kind of baseline or background. In comparative genomics, scientists use synonymous sites as neutral references to identify constrained or functional regions of the genome. And in cancer genomics, synonymous mutations have been labeled as passenger mutations. They're just hitchhiking, and they were used to build baseline mutational models. And from there, researchers derive like lists of cancer driver genes, which has guided the cancer research for the past 2 decades. Um, so our question is, what if this assumption is wrong? And this isn't just speculation because synonymous mutations, especially those that cause splicing alterations, they have been found to impact cancer treatment outcomes. So there's a striking discovery from a recent study of the KRAS gene in non-small cell lung cancer, and they found that there's a synonymous mutation G60G was found to co-occur with a neighboring missense mutation, and that caused resistance to osimertinib. And in Cell, scientists validated that when it's just the missense mutation, they were sensitive cells, but with the synonymous mutation, they are resistant cells. And mechanistically, it turned out that the synonymous mutation, it actually disrupts a cryptic splice donor site that is created by the neighboring missense mutation. And that rescued a non-functional transcript isoform, and that drove the drug resistance. So, on the surface, this is a really powerful example of how a silent mutation can alter the protein structure and function and alter the phenotype. And it's not even a mutation at the splice site, it's still embedded in the coding sequence. But the fact is, it's still alter amino acid sequence. So, could this assumption of synonymous mutuality be wrong with such rare evidences that synonymous mutations can affect the splicing and leads to disease? Um, but now here's where things get really interesting. A study in 2022, they made headline by claiming that 70% of the synonymous mutations are deleterious in yeast. It's a systematic screen in 22 essential genes in yeast, and their fitness effect were found to be comparable to missense mutations. And this really shook things up at that time, and there were they met several pushbacks, and there were scientists examining their experimental design and their interpretation of the data. So they repeated the experiment with improved controls, and they still found clear evidence that more than 70% of the synonymous mutations are not neutral, at least in yeast. So their data really conclude that synonymous mutations on a large level can have real measurable fitness effect, and that totally dispute the assumption that synonymous mutations are neutral. And this is not an isolated case. Actually, around a decade ago, studies on ribosomal proteins have shown that synonymous mutations can be just as deleterious as non-synonymous mutations. And there are also researchers found that some synonymous mutations in a glucose transporter gene, they can be highly beneficial. And there's actually a growing body of evidence across different genes in bacteria and yeast, suggesting that synonymous mutations can have fitness effect. Um, so, but the question we observe is that all of those evidence comes from prokaryotes and lower eukaryotes. And our question is, what about in human? Because our lab is majorly focused on cancer and human diseases. And despite the rare splicing or turing synonymous mutations we saw in the last two slides, um, we were thinking, are there more evidence that they're not neutral? And how many of those synonymous mutations could be not neutral? And so we think it's really critical and worthwhile to test this hypothesis in human. And that's the question we are answering today. So to answer this question, we need a system. First, we want to make precise point mutations. We want to make precise point mutations and better in the genome. And second, we want to scale it. We want to screen them at scale. And that brings us to a relatively recent innovation of a variation of CRISPR-Cas9. It's called base editing. So not like the traditional Cas9, which makes double-strand break and randomly insert or delete a fragment of gene. The base editors actually, it's a very large fusion protein, and they use Cas9 nickase that only cuts one strand, and it is fused to a DNA deaminase. And in particular, there are cytidine deaminase that converts C to T, and adenine deaminase converting A to G. And in a pooled screen, we can deliver a library of sgRNAs into a specific site. And each sgRNA can direct the base editor to a specific site, and that create thousands of targeted variants in parallel. We can then sequence and track the abundance of each sgRNA over time. So if an sgRNA drop out, it means that the cells carry that mutation is depleted and the edited mutation is deleterious to fitness. So by analyzing the dropout pattern, we can infer which mutations and how many mutations that are predicted by the sgRNAs have measurable fitness effects. And those sgRNAs that have significant fitness effects are called hits, and the proportion of hits across the group of sgRNA is called hit rate. We didn't invent the screen, but with this system in mind, we can see what previous base editor studies have found when it comes to synonymous mutations. So we reanalyzed the data from a foundational base editing study, and that study screened 50,000 clean var variants, which are potentially pathogenic, and 3,800 genes in 5 different cancer cell lines. And among those target genes, they also included 10 essential genes. So any loss of function mutations in those genes would cause sgRNA depletion, which means the cells are dead. So in their analysis, not surprisingly, synonymous sgRNAs are used as negative controls, alongside with other sgRNAs like those ones that do not make any edits in the same essential genes, and those sgRNAs that only make intronic mutations. And here's what we found after the reanalysis, is that across all 5 cell lines, the median hit rate for those negative controls were surprisingly high, including the synonymous mutations, and all of them have a median hit rate bigger than 10%. And the missense sgRNA hit rate is just a few percentage points higher than the negative controls. So, okay, when we look at this data, we were thinking, does that mean more than 10% of the synonymous mutations are actually loss of function? If that's the case, then what about those no edits sgRNA, which shouldn't have any mutation at all? So we thought about this, and we believe this observation reflects some technical limitations. And that limitation probably is about the screen, but also that limitation really limits our ability for targeting synonymous mutations. And that brings us to the next part of the story, is that these experiments weren't designed with synonymous mutations. So how would we design a base editor screen that targets synonymous mutations? And to do this, we first identified some technical challenges that can lead to the false positive signals we saw in the previous slide. So first, most base editors are designed to make edits within a very narrow window. It's around 5 nucleotide. But in practice, the edit can occur outside of the window. So even if an sgRNA is predicted to cause a synonymous mutation, it might be causing like unintended mutations nearby. And that explains the fitness effect we see. And this is actually common, and it's called bystander mutations. To solve this, we take a wider 15 base pair window when we predict the consequences of sgRNAs. So it covers the entire known editing efficiency distribution across the protospacer, and this allows us to be like much more confident that our sgRNA is only making synonymous mutations and there's no bystander. And the second issue we identified is that the base editors are very large fusion proteins and they bind the DNA. That means they can physically interfere with the transcription. And this is not random speculation because the CRISPR-mediated transcriptional regulation system is invented based on this principle. But in put screen, this is not what we want because that means the dropout phenotype might not be due to the mutation itself. So to address this, we designed a ghost editor control. We call it ghost editor because it is basically a base editor, but there's no deaminase domain. So it will bind the same DNA target, but it doesn't make any edits. And this led us to separate the transcriptional effect from the mutational effect. And finally, the third point is that the deaminase enzyme itself can sometimes like act off target. It can expose the single-stranded DNA elsewhere in the genome. And this leads to random background noise in the data. And to deal with this, we take several precautions. We perform the biological replicates in our screen, but most importantly, we do single sgRNA validation for all the hits we identified in our screen. And this is to directly confirm that the observed phenotype, it does not correspond to like random off-target effect. And all those improvements help us build a much cleaner and much reliable framework for identifying true functional synonymous mutations. So let's talk about the actual experiment. We started with over 22,000 protein coding genes across the whole genome because we don't want the experiment to be biased. And we searched for all 15 base pair editing windows that contain an NG-PAM, so it's really flexible and it covers a lot of the region. And we search for those windows that only generate synonymous mutations. That means windows like this will be kicked out. And this gives us an initial pool of 240,000 windows across the genome. And then from there, we selected a final set of around 5,600 sgRNAs, and those sgRNAs are located in genes that are strongly essential in almost all the cell lines. And those sgRNAs are predicted to make around 8,000 synonymous mutations. Alongside with those synonymous sgRNAs, we have a wide range of controls. We have positive controls, which are missense sgRNAs, and start codon disruption sgRNAs, and those ones are called iSilence. They are usually used as benchmark in this kind of base editor screen for examining editing efficiency. And we have negative controls, which are empty window sgRNAs, those that do not make any edits in the same essential genes. And we have sgRNAs that target non-essential loci and and sgRNAs that do not target anywhere in the genome at all. And we ended up with two libraries. We have around 4,500 sgRNAs for the AB library and around 7,000 sgRNAs for the CB library. And on top of that, we used the two very efficient base editor, ABAE and the CBD, which are proved to be a few hundred times more efficient than the previous generation. We ran a screen in two cell lines because we are not interested in cancer cell context-dependent effect. So we really want to find the true functional mutations. And we introduce the sgRNA library into the cells, and after selection, we collect our first baseline cells at day 0, and then we introduce either the base editor or the ghost editor. And after 12 days, which also contained another round of selection for the base editor cells, we collect our first endpoint sample at day 12. Note that the cytidine base editor is actually a little bit less efficient than the adenine base editor, so we extended the CB screen for an extra 2 weeks in K562 just to let the editing events to accumulate. And after that, we sequence the sgRNA and we calculate the fold change, quantify the abundance change of the sgRNA across 2 different time points. And we did also do statistical tests based on DE-seq2. So our first question is the screen reliable? Can it detect non-fitness consequences? And to answer this question, because most of the sgRNAs in our library is expected to be neutral because most of them are synonymous sgRNAs. So we rely on positive controls to assess the quality. And in particular, we use I-silence sgRNAs. So take the ABE screen as example. We observed that in both K562 and the Jerka cell line, the log2 fold change of sgRNA dropout, they are highly correlated across biological replicates shown as the X and Y axes here. And this means we have reproducibility across the replicate. And additionally, we observe a lot of hits for the I-silence. The hits are the red dots here, and x-axis, this is a volcano plot. X-axis is the fold change, and y-axis is the transformed p-value. And the hit rate, which is the proportion of the red dots out of all of the dots, they are around 30% for the 2 cell lines, and this is in line with the reported editing efficiency of the AB system in the current literature. So in contrast, for my negative controls, here I show the no added sgRNAs. In K562, we found that we only observe noise between 2 replicates, and there's no significant depletion for any of those sgRNAs. We do observe some, like a few spurious hits in the negative control, but overall that's like non-comparable to the I-silence library. So that gives us confidence that we're well positioned to detect the real fitness effect from the synonymous mutations. So let's dive right into the result. Are there synonymous hits? Because we have 2 different cell lines, and we first develop a stringent filter for identify the true functional hits. It cannot be a ghost editor hits, so it should not show up as a candidate in the ghost editor screen, and it needs to be significant in both cell lines. And we are not expecting like a huge depletion from the synonymous sets, so it's possible that if there's no candidates in the first time point, we will look at candidates at the extended time point. And as the result, here we showed transform the p-value in K562 and Jerkat for the X and Y axes, and the sgRNAs that passed those filters are colored red. So the larger dot will mean that the sgRNA have a stronger fitness effect. And this is in AB screen synonymous mutations. We identified 4 significant hits, and one of them are actually 5, but one of them is a ghost editor hit, so we excluded them. And in a CB screen, for the first time point, we didn't observe any hits. And for the extended time points, we found for significant synonymous hits too. As expected for the missense sgRNAs, we identified more hits, and also they obviously have a much larger effect size. So overall, we identified 8 synonymous mutation hits and 13 missense mutation hits across 2 screens. And the next question is, are those hits actually real? Could they be random? Because as I mentioned before, there could be random editing. So we follow up with single sgRNA validation assay. And in this setup, we first make the cell population that express the base editor or the ghost editor as a control. We then introduce specific sgRNAs into the cell, and those sgRNAs are tagged with GFP. So tracking the percentage of GFP positive cells over time would give us information about whether that sgRNA is deleterious or not. And here I am showing an example depletion curve for my synonymous hits in the CBE screen, and we see that the blue ones are the actual hits we want to validate in the base editor screen, and one of them is depleting, and we have positive and negative controls here too. So for all the hits, I made a heat map showing that summarizing the depletion effect across all 3 biological replicates between basically the first time point and the last time point. So basically, the darker blue means that they have stronger depletion. And for the synonymous sgRNAs, only this one showed significant depletion effect. All the other ones are false positives. And for the missense sgRNAs, most of them are validated, and there's only one false positive. So overall, this gives us our first message is that the true positive hits for synonymous mutations for synonymous sgRNAs is kind of low in the pooled screen. But this doesn't tell the whole story, because at this point, we still don't know whether the intended edits are actually made. So what if these darker blue dots are actually an effect from a random off-target mutation that we are not going to make? So that's why, and our final goal is to quantify the functional synonymous mutation proportion out of the synonymous mutations we screened. So what we did is we did extra validation on which is targeted deep sequencing at the endogenous loci. And the short answer is that yes, all the intended edits are made, and it is the predicted mutation that is causing the effect. And our next question is that with this information in hand, we can now estimate the actual proportion of the functional mutation in our screen based on the predicted number of mutations that we screened. So we calculated that in synonymous screen, we only have 1 hit, and that 1 hit, sgRNA hit, was sequenced to be 1 single synonymous mutation. And that leads to us 1 positive hit in like about 8,000 synonymous mutation site screen. And that gives us a percentage of 0.016. And for the non-synonymous mutations, those sgRNAs have 70 hits that is potentially causal to the deleterious phenotype out of 800 sites we screened, and that gives us 1.93%. And this difference is actually more than 150-fold difference. So that means the non-synonymous mutations are 150-fold more likely to cause fitness effect. And to really nail this down, we modeled the data disproportion using a binomial likelihood framework, in which this proportion we calculated is just the maximum likelihood estimates. And under the null hypothesis that the synonymous and non-synonymous mutations have the same probability of causing fitness effect, we calculated the likelihood ratio, we conducted the likelihood test, and we visualized the likelihood surface across a range of synonymous and non-synonymous probabilities. So as you can see that the X and the Y axis scale is actually really different for visualizing this peak. That means they are far off the diagonal, which is where the null hypothesis lies. And we can see the highest point, which is our observed probability, is highly significant with a super small p-value. And that means that this provides a quantitative evidence that while synonymous mutations can matter, we observe um, one significant synonymous hits, and we actually have extra evidence, but I'm not showing it here. But even though we observe one synonymous hits, but they are actually less substantial to impact the cell fitness than non-synonymous mutations. So our take-home message is that they are mostly neutral in human genome. We don't know how they do it in the yeast, but in human, we have solid evidence now. And in conclusion, we developed a genome scale high resolution CRISPR base editing assay to measure the fitness effect of synonymous mutation. And that's really rigorously controlled for synonymous mutation. And we found that most of the synonymous hits are false positives. But even though we do know that the false negatives in our screen is a consideration, we estimated that the functional synonymous mutations are much less likely to impact fitness than non-synonymous mutations. And our work, despite that, despite of all it's kind of negative result, but it highlights the importance that we should pay more attention to the experimental design and proper controls in this kind of pooled screens that measure subtle variant effect. And that's it. And I have an ad that I'm graduating in spring, so I am looking for a postdoc position. And this is an experimental work that I am doing, but I also work on computational biology projects. I worked on identifying synonymous cancer drivers, and also I am in the collaboration project that we study complex genetic interaction in kinase phosphate binding loop. And I am interested in cancer research, proteomics research, and synthetic biology research. And this is my email. So thank you. Any questions?
All right, thanks, Yiyun. That was a really interesting talk. So just a reminder, if you have any questions that you would like to ask, put them into the Q&A and I can share those. So our first question is from Katie, who asks, do you have any ideas about how the single synonymous hit that you found, how it is impacting fitness?
So we are looking to that, and that is one of the future direction. But one possible mechanism we observe is that it seems to disrupt the binding of one of the splicing factor, and that is one of the direction we are looking into. Yeah.
Cool. And then our next question from Sven asks, did you have the chance to check whether any of your over 8,000 hits would target splice sites in essential genes and then, would have actually been seen within the assay? Like, I guess where are those
Yeah.
edits kind of targeted or placed around those genes?
That's a really good question. And when I designed the library, I actually explicitly avoid those synonymous mutations that locate at the splice site. So they are strictly in the inner part of the coding region. And yeah, that's one detail that's I didn't mention, but it's important.
Great. So I guess I have a question. Um, Looking back at or I guess have you looked back at the yeast data to see whether or not there's kind of patterns in the synonymous mutations that they're testing? Like, I guess in line with the previous two questions, like, are there kind of mechanisms that might explain the really stark differences in results that you see?
Yeah, so in their paper, their major argument, even though they don't have a very systematic mechanism, they didn't come up with a systematic mechanism, but it's majorly about the mRNA expression level. And I think that's possible I think that's possible, but since most of my candidates are negative, so we didn't go into the direction of or whether the expression levels are changed. We're more interested in that one single hit, and that doesn't seems to be the expression level. But yeah, they are But in that East paper, they didn't talk more, like because the expression level expression level change could be caused by many things like mRNA structure or its stability, and then affect its half-life. But yeah, they didn't provide more evidence for that.